and good afternoon, everyone. I am Jeremiah Perez, and I shall be presenting my thesis entitled An Immunoinformatic Approach to Developing a Possible mRNA Vaccine Construct Against Respiratory Syncytial Virus. Respiratory Syncytial Virus, or RSV for short, is an orthopneumoviridae virus first discovered in 1956 that causes lower respiratory infection in infants and children. What makes this virus dangerous is that it displays harmless symptoms similar to that of the common cold during its first few stages, but can develop into significantly worse conditions that can have debilitating effects on infected patients. As high as 50% of affected patients and children and 25% of affected adults may contract bronchiolitis as a complication in progressing RSV infection. Children who have experienced severe symptoms often live with additional long-term complications like asthma and wheeze. RSV infections are not restricted to children and will affect nearly all ages, especially the more vulnerable older patients and the immunocompromised. The vulnerable demographic is in fact at greater risk of acquiring possibly life-threatening health conditions like pneumonia and bacterial co-infections. Preventive and supportive treatment methods against RSV exist but are only successful to a limited degree. And as of late, there are no licensed vaccines against RSV. There are groups that have been working on creating a vaccine, like the company Novavax, but as of 2018, they have yet to clear phase 3 of clinical trials. Given the severity of symptoms and the stagnation of the vaccine development process at the clinical level, an immunoinformatic approach must be considered for developing a vaccine. So, of the 11 proteins in RSV, the fusion protein presents itself as a viable target due to its accessibility to neutralizing antibodies and its relatively more conserved nature compared to the G protein in terms of frequency of amino acid variation. Additional studies suggest that the A subtype poses a larger threat due to a greater risk of developing severe complications as seen in higher likelihood of hospital admission for children infected with RSVA variants. With that said, an mRNA vaccine containing the sequence of the F protein shows the most promise for subsequent buildup of immunity against RSV. The primary objective of this study is to identify the T cell and B cell epitopes of RSV via an immunoinformatic approach in tandem with population coverage studies and in silico simulations against a human immune system model. This is done to assess the efficacy of the epitopes in the vaccine construct. It is worth noting that the study will only be dealing with the creation of an mRNA vaccine derived from the F protein of the S2 strain of the RSVA variant. The study has three phases: epitope identification, the development of a vaccine construct, and in silico simulation against the human immune system model. Flashed on the screen is the first phase of the study, which is the epitope identification phase. Oops. So the FASTA sequence was first derived for RSVS2 strain, and after which the subsequent identification of epitopes and their MHC class 1 and class 2 allelic partners was done with the necessary resources. So for my case, I used NetCTL right here for CTLs, IEDB for HTLs, IBCEL for LBLs, and for allelic partnering, I only did it for the CTLs and the HTLs. Once the epitopes have been identified, toxicity, immunogenicity, antigenicity, and cytokine inducibility studies were done where applicable to narrow down the most viable epitopes. The second phase of the study is the creation of the vaccine construct. All of these viable epitopes were subjected to multiple sequence alignment and population coverage studies to determine any mutations and assess the efficacy of the epitopes against the global population. After this, an adjuvant epitope vaccine was established as the preliminary vaccine construct. A final screening of the vaccine construct's antigenic, toxicological, and allergenic properties was done, followed by the simulation phase. Now let's go over briefly the results of the study. Flashed on the slide is my protein of choice, which is the F protein. This is a class 1 fusion protein that requires proteolytic cleavage to present the fusion component that is buried deep within the structure. It is a metastable protein that is anchored perpendicular to the surface of the cell, or viral membrane rather. The function of this protein is to mediate membrane fusion between the target cell and the virus. So next is my compilation of results for CTL, HTL, and LBL epitope prediction and assessment. After rigorous selection and physicochemical property testing, these are the peptide sequences that I considered as viable epitopes for CTL, HTL, and LBL. For CTL, I first found 210 MHC class 1 responsive peptide sequences. For HTL, I found around 150 or so MHC class 2 responsive peptide sequences. And for LBL, I found 36 B cell responsive peptide sequences with probabilistic scores of 0.75 or greater. Subsequent physicochemical testing yielded 9 viable epitopes for CTL, 2 viable epitopes for HTL, and 5 viable epitopes for LBL. All people possess sets of MHC class 1 and class 2 genes, which offer a wide range of specificity in terms of MHC peptide binding. Given the polymorphic and polygenic nature of MHC molecules, it is important to determine the MHC allelic partners, as you can see right here for uh, CTL and HTL right here. It is important to determine these allelic partners for each corresponding peptide. So allelic partnering scores were considered at percentile ranks of less than or equal to 1% for CTL and less than or equal to 10% for HTL, as sequences with these scores are said to have the highest binding affinity and therefore have a better chance of inducing T-cell mediated immune responses. Cytokine inducibility is also a major consideration for choosing possible HTL epitopes, since some cytokines aid in modulating adaptive immune response. For this case, I chose the IFN gamma, IL-4, and IL-10. Next up is our studies for the MSA, which is used to determine the extent of evolution of the RSVF protein, and consequently the possibility of using these epitopes derived from S2 strain against other variants of RSV. As seen on the results right here, you can see that there is a great degree of mutation for this particular epitope alone. All 16 epitopes were tested against 243 randomly selected F protein sequences. And of the 16 epitopes, only 9 were present in at least 83%, as you can see in the results right here. The reasons for the absence of the epitopes is associated with the sheer number of known RSV clades, as you can see on the left. In total, there are around 38 known clades of RSV, as well as the additional, or rather, the high rate of nucleotide substitution in RSV. So for context, the S2 strain is located somewhere around here, and that only accounts for a very small portion of all the known strains of RSV right here. In any case, the vaccine construct should still work, barring the 7 other absent epitopes, since these peptide sequences have been established to be immunogenic and antigenic. Next is my results for the population coverage studies, which was done to determine the possible coverage of the vaccine relative to dominant HLA alleles per region of the world. A mean global coverage of 92.8% was established, with Central America having the lowest population coverage at 33.4%. So the reason for this disparity was attributed to HLA allele polymorphism, since polymorphic genes are known to prevent certain MHC class 1 and class 2 alleles from responding normally. Next is the creation of the vaccine construct, which was done with all 16 epitopes, the linkers, and an adjuvant. For my case, I chose the CD40L as an adjuvant because it's already an existing co-stimulatory molecule found at the surface of T cells. In addition, its adjuvanticity has been verified in mouse models, as shown in results for improved T cell response, and therefore improved chance of immunization. For the linkers, I used GPGPG for adjuvant and HTL links, AYAC for LBL to LBL and HTL to LBL links, and AAY for CTL to CTL and LBL to CTL links. The results of the vaccine construct's physicochemical property testing yielded favorable outcomes, as noted in the acceptable standards for the results of amino acid scores, or rather amino acid numbers, and the molecular weight of the vaccine. The aliphatic index and the instability index also suggest that the protein is stable and has some degree of resistance to high temperature. Charged residues are also known to aid in membrane insertion and formation of RNA protein complexes, and the presence of these uh, charged residues may increase entry efficiency into the cell, and therefore increase epitope affinity to respective proteins. The following sets of slides are the results of my immune system simulations. As expected, the vaccine construct follows known immune response patterns based on key immune cell types, as well as showing other trends indicative of successful immunization. First up is that the vaccine construct shows a decreased likelihood of causing autoimmunity, as seen in the high IL-2 and low B signal concentration. This suggests that the epitopes promote an immune response with a general aversion to cellular distress. In addition, the low concentrations of IL-4 and IL-10, right here, the smaller graphs, are indicative of a sufficiently modulated immune response without any overactive inflammatory cytokines. Next up is the B cell response. Trends in B cell activity also indicate successful long-term immunization, as noted by the year-long persistence of B cells, memory B cells rather, and IgG and IgM producing B cell variants, as you can see in figure A. There is also year-long persistence of B cells in the active state, as you will see in figure B. Aside from the long-term immunity, B cell trends also show the possibility of isotype switching between IgG and IgM in particular, which gives an overall versatility to the immune response. That can be seen in the spikes in antigen concentrations for IgG1 and IgG2 and IgM during each instance of vaccine introduction. Next is the results for helper T cell response. The vaccine construct shows evidence of successful long-term immunization relative to helper T cell, as observed in the persistence of helper T cells for an indefinite period of time, as you can see in figure A. Moreover, some results also consolidate the successful HTL epitope recognition as seen in the activity of the TH1 helper T cell variant, as you can see right here in the purple line. Lastly is the killer T cell and other immune cell types response to the vaccine construct. There is seen to be consistent activity of other immune cell types, which indicate to some extent the safety of the vaccine against autoimmunity. This can be observed in the persistent dendritic cell activity as seen in figure B, with some notable spikes in presenting dendritic cell concentration, which can be observed in this small green line rather. That indicates its involvement in each instance of immune response to a limited degree. There is also persistent macrophage activity as seen in figure C right here, with active macrophage and presenting macrophage showing trends expected of immune response patterns. And lastly, the persistent epithelial activity as seen in figure D. The results for this graph in particular also consolidates the avoidance of autoimmunity for the vaccine construct, as seen in the absence of any notable decline for epithelial cell, meaning there is a lack of overly aggressive immune response against healthy cells in general. So to summarize my study, I have established 16 epitopes for the RSVA S2 strain with 9 CTL, 2 HTL, and 5 LBL epitopes. All of these epitopes pass prior screenings for immunogenicity, allergenicity, toxicity, and antigenicity. Multiple sequence alignment studies show that 9 of the 16 epitopes found in S2 RSVA were present in at least 83% of the 243 verified F protein sequences, while population coverage studies for the established epitopes averaged to about 92%. I believe some adjustments must be done in terms of filtering the epitopes, especially for HTLs and allelic partnering in general. Adjustments for epitope selection may also significantly improve the population coverage score. A viable vaccine construct was established with CD40L as an adjuvant, and the respective testing of its physicochemical properties showed acceptable results. In silico simulation with a human immune system model showed an overall positive response with little risk of complications like autoimmunity, therefore pointing to effective long-term immunization with a vaccine construct containing the CD40L adjuvant and the RSV epitopes. That concludes my presentation.